Hello everybody and welcome back to the Computational Genomics Lab. Today we are going to install packages in RStudio. In the previous video we installed RStudio, kind of went over the frames, and we'll keep doing that as we go. And we are going to install our first package, and then after this we are going to actually start doing analysis and we'll be done with our setup. We'll have to install packages as we go, and that's one of the great things about R is there's tons of free software on one of two websites, and it's either on the Comprehensive R Archive Network or CRAN, and that's here, cran.r project. This is where you actually installed R from, and there are tons of R packages in here, and we are going to use Tidyverse is the first package we're going to install. So it's from the CRAN repository, and the other one is the Bioconductor you might have heard of. And Bioconductor is also our software made by somebody. There's just two different warehouses, if you will, of where these packages are. And so you'll know as you go along where to get them, but you have to install them a little differently. And so let's first install Tidyverse, and just as a quick intro to Tidyverse, because it is one of the coolest things, feel free to go and look more about it, but it's just a logical way of writing your code. And so if you go to Tidyverse, you go to any package really, it'll tell you how to install it, and it's install.packages Tidyverse. Pretty simple. Okay, let's try it out here. And this might take a minute to install, but anytime we're installing from the CRAN, we're going to use install.packages function. You can see R already knows that that's the base. R knows to go to CRAN. It doesn't know automatically how to go to Bioconductor, and that's what we're going to do next. Actually, let's do that first. Since we can install directly from R, we're going to install what is called the Bioconductor Manager. And so we go back here, and we'll just Google install Bioconductor install, very easy, and it gives you the code right here. And so we're literally going to just copy and paste this, and it's basically saying if you don't already have this, go ahead and Bioc Manager : install version 3.18. And when we install other packages, we're going to see that Bioc Manager : install parentheses and then the package you want to install. Okay, so here we are. We're going to paste that in, and voila, we have already started installing our first package. And so I'm going to let that, oh, it's done running. That was fast. Nope. Okay, so now if we want to install something with Bioconductor, we can do Bioc. See, now it knows that. It didn't know that before. So it's Bioc Manager : and then install. And these are automatically filling in down here, so which is really handy as you're going along. And so now we can install anything from Bioc Manager. We're going to use a program called DE-seq for our RNA-seq analysis. That's on Bioconductor. Some of the packages like Tidyverse are on CRAN. And so let's install a CRAN package. And we'll do install. Should find it. And then you can just tab forward packages. You do need quotes for the package name. And we're going to do Tidyverse here. And so we'll just hit that. And it's going to go, this is going to take a second. And then once that installs, I'm going to show you where to, how you then load a library. Oh wow, this is going really fast today. I don't know what's happening here. So now we have Tidyverse, and so one of the functions in Tidyverse is how to read tables better, how to make a pipe, which means go to the next line of code. And we're going to go all through this very simply. For each task we want to do to analyze our data, we're going to learn the different R functions. But the goal for today is just to install packages. Okay, so we've seen that there's the Bioc Manager, and there is install.packages. So Bioconductor, CRAN, you got it. That's all there is to it. And so whenever you're writing your code, we kind of made our first R markdown file here, and we did library Tidyverse, and previously it said, hey, you need to install this, but we now have it installed. So it knew, and it fixed that error. Okay, so now when you're doing your setup chunk, this is the first chunk you'll make in any code file, and it's how to set up R. Like, what packages am I going to use? And so we're definitely going to use Tidyverse, and so we're going to run this. You can either highlight it and hit run, or you can hit control return, which is what I'm going to do. Okay, so we now have Tidyverse run. And this is a bit annoying that the results from down here came into our R markdown file. And so you can go to this little cog here and say chunk outputs in console. So it means it's not going to put it in the R markdown file where we don't really need to know that. It's going to put it down here in the R console. And I'm going to do remove output. Cool, it's gone. And so now next time we run it, we're just going to load library Tidyverse. So that's the function. Library is to load it. We did install.packages to install it. Notice the only difference here is we don't need quotes. So we did that. And so we just do library Tidyverse. Okay, so I'm going to put in the description and on the GitHub a list of packages to install, because some of them do take a lot longer than this. For example, DESeek2 is going to take a minute to install. And then as you're about to write your code here, you need to have run this start chunk to load the packages in. So you've installed them, and then you load them up here for each file. And that's kind of nice, because then for a different set of analyses, you might make a different R markdown file and then just load the essential packages for that analysis. Okay, so we're going to do one Bioc Manager install. I don't think it's going to finish in time. And so this is really how easy it is. So like I'd say we're going to really need DESeek for a lot of the things we do. So we can search install DESeek2. We're here, and it has this, if required Bioc Manager. We don't really need that anymore. Now we just need this Bioc Manager install. And so because we've installed the Bioc Manager, we can install packages with the Bioc Manager. I hope that makes sense. But like install.packages built into base R to go to CRAN, and sometimes we're going to want to go to Bioconductor, so we had to install that Bioc Manager in order to install packages from Bioconductor. It'll all make sense later. It's a little around the elbow with this one, but we can start typing in. We see Bioc Manager. I'll do tab, install, and then in quotes, I have to get this spelled right. DESeek is capital DES, lowercase eq2 for DESeek2, and hit enter, and boom, we are installing DESeek2. Now this one, no. Okay, yep, so now it's going. Whenever it asks you, do you want to update other packages and other things, always hit N for no. Otherwise, each package has other packages inside of it, and so it's saying it right now, update all, some, or none. And I'm going to hit no for none, because I want to leave my packages the way they are. And sometimes you'll have to hit yes, but I rarely would do that, because what it's going to do is go find all those other packages and upgrade them. And now your other program that uses those packages might not need that newer version. It might need an older version. So always best to hit no when it says update some, all, or none. Okay, so we got a lot of things like masked functions and things like that when we installed DESeek2, but the good news is it is installed, and the way we do that is the same thing we did with Tidyverse, is we set it up here with library DESeek and hit enter to load that library. If it just goes there automatically, it means it's all good and it knows it's there. And so some of those warnings can be scary. There was a bunch of red down there, but the best thing to do is just type it up here, library DESeek2. If it installs, then it's good. And unfortunately, there's no way to just test if DESeek's working because you have to make a data file and other things that we'll be doing shortly. But you can start typing in the function like DESeek and hit tab. And we can see it up here is DESeek. This means that R knows the package is installed, it knows the parameters, it has a bunch of information about the package, and so that means it's installed. And so, okay, so in the video description and the GitHub, I will have some other packages for you to install just so we can save time moving forward. But for now, really all we need is Tidyverse and DESeek. And then we are going to next start loading in the files that were outputted from the RNA-seq pipeline and learn some R basics slowly, and we'll get moving faster with that. But the first thing we're going to do is use Tidyverse to read in an RNA-seq table, make an object, learn about what those are, and go from there. So with that, I'll see you in the next video, and looking forward to starting our analysis. No more installing software. We are good to go. And we have tons of free packages ready for us from CRAN and Bioconductor, so feel free to search around and go ahead and install them. It's all free. All right, we'll see you next time.